Assalamualaikum and hi, my name is Nur Yasmin Soraya binti Idris. For today's video, my group will present about our poster that has been extracted from a journal titled "The Spice Effectively Ameliorated Hyperuricemia and Hyperuricemia-Induced Kidney Injury in Mice." Clove, scientifically known as Syzygium aromaticum, is a common culinary spice and an important herb in traditional Chinese medicine, where it is believed to enhance kidney function. This is relevant because hyperuricemia is now the second most prevalent metabolic disorder, where elevated serum uric acid results from increased production or reduced elimination, and persistent elevation eventually leads to monosodium uric crystal formation that causes gout, hypertension, and renal impairment. Next, current hyperuricemia medications such as allopurinol, febuxostat, and benzbromaron are effective but limited by safety concerns. Therefore, safer alternatives are needed. However, very little research has explored the uric acid lowering effects of clove water extract and its potential to prevent hyperuricemia-induced kidney damage. Thus, this study aims to fill that gap. This study hypothesized that clove water extract has strong anti-hyperuricemic effects and can significantly lower serum uric acid levels. In addition, it may regulate uric transporters by reducing UR81, GLUT9, and MCT9 for reabsorption, while increasing ABCG2, O81, and O83 for excretion. This study also proposed that clove water extract protects the kidney through modulation of inflammatory pathways and cytokine-related interactions.
Hi and Assalamualaikum. My name is Muhammad Haziq Iskandar bin Muhammad Shamsul, and I am going to explain the 4 objectives in this research paper. So the first one is to evaluate the clove water extract, knowing whether it's working or not. So the researcher wanted to know whether the clove water extract can lower the high uric acid level in the mice that had the disease. But knowing it is working is not enough, so it will lead to the second objective, which is to determine the clove water extract is safe or not. So the infected mice will be checked their body weight and their organ. So if the treatment is being given, they will know whether the toxicity is happening or not. And the third objective is to assess the clove water extract, whether it can protect the kidney or not, since high uric acid level can cause a damage to the kidney. So the researcher wanted to know whether this extract can reverse the damage or prevent from further damage. And lastly is to elucidate or to figure out how it works, the protein or biological pathway, how the extract change to flush out the uric acid from the kidney. Now we will moving on to the technology used and why they are used. So first, to identify exactly what they were testing, they used the ultra-high performance liquid chromatography coupled with a Q-exactive quadrupole Orbitrap high-resolution mass spectrometry. So this advanced technology analyzed the chemical makeup of the clove water extract to confirm its active ingredient. Secondly, they established a hyperuricemic mouse model by giving the mice specific chemical. So they created a condition that mimics the human disease and allowing them to test the treatment in a living system. Third, to measure the result, they used a biochemical assay and hematoxylin and eosin staining. So the assay provided exact number for uric acid level and kidney health, while the staining allowed them to visually inspect kidney tissue under a microscope for the damage and repair. Next, to understand the mechanism, they utilize the Western blotting. So this technique measured the protein level of specific kidney transporter. So they will show the exact how the drug help excrete the uric acid. And finally, they perform the transcriptomic analysis and real-time quantitative reverse transcription polymerase chain reaction. So this genetic tools scan the kidney cells to find the specific inflammatory pathway the drug turned off. so ensuring the finding were accurate at the genetic levels.
Assalamualaikum and good day everyone. My name is Auni and today I will be presenting the key findings of a study investigating how clove water extract can be used as a natural treatment for hyperuricemia. The context of this study is simple. The current standard drugs for high uric acid can cause severe side effects including allergic reaction and cardiovascular risk. Therefore, we need a safer alternative. In this study, the key discovery is that clove water extract (CWE) is a highly effective dual-action agent against hyperuricemia and associated kidney injury in mice. Now, let's look at what the researchers actually found. First, CWE significantly lowered the serum uric acid in hyperuricemic mice. Second, it also protected the kidney by reversing the damage and improving the kidney functional markers like creatinine and urea nitrogen. This is also supported by a study from Abdul El-Aziz et al. 2024 that reported there is a reduced inflammation and oxidative stress in kidney tissue. Third, CWE also demonstrated that there is no signs of toxicity, which is a very excellent safety to the major organs. Next, how does it work? This mechanism shows a triple action strategy which can be summarized into IER: inhibit, enhance, and reduce. The first one is inhibiting production. CWE acts as a natural drug that can suppress the activity of the enzyme xanthine oxidase, XOD. This is the brake of the system which can reduce the initial synthesis of the uric acid. Second, enhancing elimination. It suppresses the reabsorption transporters like URAT1 and GLUT9, preventing uric acid from being recycled back into the blood. On the other hand, it increased the elimination transporters like OATs and ABCG2, ensuring more uric acid is actively pushed out through the urine. Finally, reducing inflammation. CWE acts as a damage control. As we all know, high levels of uric acid can cause kidney inflammation. But this extract was found to modulate the cytokine-cytokine receptor interaction signaling pathway, thereby reducing the inflammatory signals that lead to the tissue damage. In short, clove water extract (CWE) fights high level of uric acid, clears it from the body, and calms the inflammation it triggers, all naturally and safely.
The novelty of this study is that it is the first comprehensive evaluation of clove water extract for hyperuricemia using biochemical, histological, transporter-based, and transcriptomic analysis. It was found that low-dose clove water extract has potent urate-lowering effects without organ toxicity. The study also identifies modulation of major urate transporters and reveals 38 hyperuricemia-related genes reversed by clove water extract, including key susceptibility genes. Additionally, clove water extract shows anti-inflammatory and nephroprotective activity through transcription factor and also chemokine regulation. This research highlights clove water extract as a safer alternative to conventional hyperuricemia drugs. It scientifically supports the therapeutic value of edible medicinal plants and bridges traditional Chinese medicine knowledge with modern pharmacology by showing clove's ability to regulate renal urate transporters and inflammatory genes. All in all, the identification of bioactive compounds such as bifluorine, isobifluorine, and azelaic acid provides a foundation for developing new natural anti-hyperuricemic drugs.
Hello, as the final presenter, I will now go through the strengths, limitation, and the overall conclusion of this study. Firstly, the strengths of this study. One major strength is that the researchers use a wide range of scientific methods, including biochemical tests, kidney histology, Western blotting, and transcriptomic analysis. By combining all these methods, the study quite provides a strong and reliable evidence showing how the clove extract works in reducing uric acid and protecting the kidneys. So another important is that Another important strength is that the clove extract showed multiple beneficial effects. It didn't just lower uric acid levels, it also helped repair kidney damage, reduce inflammation, and improve overall kidney function. This makes these findings more meaningful because it demonstrates that clove has more than just one single action. It offers a broader therapeutic benefit. As for the limitation of this research, the first major limitation is that the entire experiment was conducted only on mice. While the results are promising, we still cannot be certain that the same effect will happen in humans. So more clinical trials are needed before clove extract can be recommended in real medical settings. The second limitation is that although the researchers identified several active compounds in the extract, they did not determine exactly which compound is responsible for the uric acid lowering effect. This means that mechanism is only partially understood and more detailed studies are needed to confirm which compound is truly effective. Finally, the conclusion of this study, the research showed that clove water extract can effectively lower uric acid level by reducing its production and increasing its excretion. It also provides strong kidney protection, helping reducing inflammation and preventing tissue damage in hyperuricemic mice. In addition, the extract appears safe with no significant toxicity or harm to major organs. Overall, this study findings suggest that clove could be a promising natural option for managing hyperuricemia. However, because the study was done in mice, human research is still required to confirm its effectiveness and safety. So that's all from me. Thank you.